Good day, grade 12 learners, and a very warm welcome to our wonderful grade 12 educators. Welcome to today's lesson, Life Science subject lesson. My name is Samantha Ragunanan. This lesson is brought to you by Gauteng Department of Education in collaboration with Saibono Discovery Center. Let's begin. First, we're going to start with the exam guideline. Now, grade 12 learners, this is a very important document. I cannot stress this enough. So what an exam guideline basically is, you know how your textbook's got so much of information? It's so much to learn, but we've made it easier for you. We've given you a little recipe for success, and this is called our exam guideline. So this exam guideline is given to the examiners who set the exam paper, and they are told these are the topics that you set the exam on. Isn't that so easy? So now you can remove all those big chunks of information from the textbook. You can streamline what you are studying and zone in on all the pressure points. So let's go through the list. Now, today we're going to discuss meiosis. Meiosis is in paper 2. It consists of 21 marks at 14%. So this is a smaller section. Grade 12, this is the section where you can get the marks. This is a section that is easy to understand. There are so much of beautiful diagrams that you can remember, stories to tell in each one of the cases. Wait and see, I'm going to show you all. So let's go through the list very quickly. You need to differentiate between replicated and unreplicated chromosomes. This is where terminology comes into play. You know, all those terminology tests that your grade 12 life science educator has been given you, that is very important, okay? Because life science is made up of its very own language, and we need to use this terminology in the exam. The next point is discuss the different stages of meiosis. So meiosis is made up of different stages. You need to compare meiosis and mitosis. Mitosis sounds familiar to you, right? You did that in grade 10. So we're going to touch on that just a little bit. And then we're going to differentiate between meiosis 1 and meiosis 2. We're going to discuss non-disjunction and Down syndrome. So that is the list of topics for meiosis for this week. So let's discuss, where does meiosis take place? Meiosis takes place in both plants and animals. In animals, as you can see, it takes place in the testes in the male. So it takes place in the testes, and it takes place in the female. It takes place in the ovaries. Okay, so we can already see it takes place in the gametes, isn't it? In the sex cells. And then it also takes place in the plants. So let's look at where it takes place. So it also takes place in the male and the female counterpart of the plant. So in the flower, it takes place in the anther. There's the anther, this diagram there. And the ovules, we recognize the ovules, isn't it? Because it looks like a lady's pregnant belly. That's how I always remember, stigma style ovary, and the ovules are found inside there. So that is where meiosis takes place in the plants, in the flower. Moving on. So there's the ovary and the anther. So let's recap on mitosis, our grade 10 mitosis. Now, what you need to understand about mitosis and meiosis is that the only way you're going to learn this section is with diagrams. And the diagrams themselves tell a story. It starts with a certain number and it ends with a certain number. Very simple. If by the end of this you do not understand mitosis and meiosis back to front, then I don't know. But you are definitely going to get those 21 marks, okay? So let's start. Let's talk about interphase. So interphase, we know that interphase is not part of the cell division process, but it is a very important process. As you can see in the diagram, there is a whole lot, a bunch of chromatin network there. And what is happening there? We know what's happening there. We've just come out of that section. Am I right, grade 12s? Right, DNA replication. Okay, we're going to touch on this very briefly because I know you know your grade 10 work. So there we go. We've got interphase, and interphase, the DNA, the chromatin network is there, the DNA replicates itself, and then we move on to the next phase, which is prophase. So prophase, as we can see from the key that we have there, we have the maternal chromosomes and the paternal chromosomes. And this is where prophase is actually where the chromosomes become distinct. We can start to see them, we can see their shape, we are able to identify them. Whereas before this, they were in a tangled mass. Okay? And then the next phase, metaphase. We remember this. Remember your mnemonic, meta, M for middle, isn't it? So there, the chromosomes are aligning at the equator. They're aligning at the equator, and we go on to anaphase. Anaphase is where they are moving away. A for away. So they're moving, the chromosomes, the chromatids, they split up and they move to the opposite ends of the pole. Can you see that? And then we move on to telophase. So telophase is T for 2. The cell divides into 2 and is the identical to the parent cell. If we can call this the parent cell, it is identical to the parent cell. If you count the number of chromosomes, 1, 2, 3, 4, 1, 2 from the maternal origin, 1, 2 from the paternal origin. So we can see they have formed a carbon copy of themselves, like a rubber stamp, hey? Now, in order to remember all these things, remember, life sciences has a lot of facts. It's got a lot of notes. So we have to find ways in which we can remember it. And if you don't know this mnemonic as yet, you should learn it. It's called IPMAT. So IPMAT, you can even make a sentence with these letters. We take the first letter from each phase. So there, I for interphase, P for prophase, M for metaphase, A for, yes, anaphase, and T for telophase. So what we basically do is we take the first letter of each one and we form what is called a mnemonic. And when we are in the exams, you don't have to remember all these notes. We remember our mnemonic, we picture the diagrams in our mind, and we know the process of mitosis. So from our grade 10 work, we know that the purpose of mitosis is basically for growth, for reproduction, for repair, and also to replace damaged cells, because our cells get damaged and they need to be replaced. So because it creates an identical copy of itself, we are able to replace that damaged tissue. Okay? Let's move on. So, on meiosis, meiosis undergoes a replication process. It's basically a reduction division. What happens here in meiosis? You have DNA replication which occurs there. Okay, so you have a diploid cell. This diploid cell is made up of cells from the maternal and the paternal origin. And the cell duplicates itself through DNA replication. So when it duplicates itself, it is still the same ploidy, it is still diploid. But then it undergoes another cell division. And as you can see, from diploid, it changes to haploid. So diploid meaning di, meaning two sets of chromosomes. Haploid, half the number of chromosomes. So we know that in meiosis we have 2 division processes taking place. Okay, that was the first division. And then the second division takes place in the very same way. You have the nucleus, you have the single set of chromosomes, and they divide further to make another 2. Okay, and as we can see, we start off with 1 cell and we end up with 4 cells. And the definition of meiosis can be summarized in just this diagram. You have a parent cell which starts off as 1 cell and it undergoes 2 divisions to form half the number of ploidy as the parent cell. Okay, so we're going to take a 2-minute break, just a little bit of a breather. You can grab some water. It's a very hot day today. and then we'll continue in just a minute. Thank you. And we are back, grade 12s. Welcome back. The next part of our work, we're going to discuss the difference between haploid and diploid. So we know that every species have a set number of chromosomes. In humans, we like to talk about humans because it's easy for us to relate to. So humans have 46 chromosomes, which is 23 pairs of chromosomes. Every species has a specific number of chromosomes in the nucleus. So if we look at the difference between haploid and diploid, we look at haploid. Haploid has just one set of chromosomes, and the chromosomes are unpaired. Okay? In the example, in humans, we have they're called sex cells or gametes, and we know them as sperm cells and ova. And they only have 23 chromosomes. Remember, they have 23 because they are half of a full set. The full set that we have is 46 chromosomes. So if we go to diploid, we can see that it has double the set of chromosomes. So if haploid had 23, then obviously diploid is going to have 46 chromosomes in humans. Our somatic body cells have 23 pairs of chromosomes, which is 46 chromosomes. So we need to be careful here, because very often if they ask in the exam paper, they ask about the number of chromosomes. So you need to pay attention and find out, what are they really asking for? Are they asking for the pairs of chromosomes, or are they asking for the numbers of chromosomes? So we need to distinguish if they're asking for the pair of chromosomes or the number of chromosomes. So we have to read our question very, very carefully. Okay? And then we have a beautiful little animation here of the sperm fertilizing the ovum, and classic example of a difference between a haploid and a diploid. A female gamete, there's the female gamete. The female gamete is haploid, meaning it has half the number of chromosomes. We have the male gamete, which has also half the number of chromosomes. And if we add them up, we will get a diploid number of chromosomes, which results in a zygote. Okay. Let's get on to our first phase of meiosis that we are going to discuss. So when it comes to meiosis, meiosis starts off with interphase. And as we mentioned earlier on, interphase is not part of the division process. It is not. but we feature it in the beginning of mitosis and in the beginning of meiosis, simply because it is a very, very important phase. Because if the DNA doesn't double or replicate itself, then there will not be enough number of chromosomes at the end. And this is important for the continuation of the generations. Okay? So let's talk about interphase. The DNA replication occurs. So we remember what happens during DNA replication, am I right? The double helix unwinds, the hydrogen bonds break, they unzip, and the DNA molecule forms a duplicate of itself. It replicates itself. So now we have two DNA molecules formed. So now we're going to distinguish. So DNA replication helps to double the genetic material to be shared equally during cell division. So let's distinguish between a unreplicated chromosome and a replicated chromosome. So we're going to do the definition first, because it's important to read with understanding. So whenever you come across a term that you don't understand, read the sentence, then read it with understanding, underline the key words, and then draw a little sketch, because we learn better with diagrams. So we're going to try and practice that now. Okay, so let's do unreplicated chromosomes. It is a single, already we can picture a single line in our mind, am I right? A single chromosome as it appears in the cell before DNA replication. And then the replicated chromosome. The replicate chromosome, it's a double chromosome with two chromatids after DNA replication. So already in your mind, you can envision just from the definition, when something is replicated, it is copied, isn't it? When something is replicated, it is copied. So if we have one copy and if it replicates itself, we are having an identical copy. So let us look at what that looks like in a diagram. There we have it. We have an unreplicated chromosome. Can you see the purple structure there? That is an unreplicated chromosome. It is on its own and it is single. And then what happens? On the right hand side, we have an example of a replicated chromosome. So that is a double chromosome. Can you see there's one, there's two. So the unreplicated chromosome replicates and it forms a double chromosome. And if we learn these diagrams, if we draw these diagrams along with learning the definition, so it's not just enough. Yes, we know that we learn better with diagrams, but it's not just enough to know the diagram. We need to know the terminology that goes with the diagram. Okay? So that is interphase, the replication process. If we move on, we're going to discuss what is a homologous pair of chromosomes. So a homologous pair of chromosomes, when we don't understand a definition, we try to break the word up. As in any subject, we break the word up. So homo meaning the same, and logos meaning the gene position of the chromosome. So homologous chromosomes are identical. They are identical. So if we look at the diagram, you're probably wondering, you know what, we know that they're identical and we just need to be able to identify them. No. In past papers, it has come out where you needed to understand the characteristics of what is a homologous chromosome. Okay, so let's look at each one. And we picture it in our mind, we look at the diagram and we try and put the words with the diagram. So let's go to it. Right, they must be the same shape. Now look at these two examples that we have here. Okay, they are the same shape, they are longitudinal, and they are of similar length. Can you see? They are of similar length. So their length is similar, and they're actually supposed to be almost identical in size. And then the position of the genes or the alleles. If you look at the alleles are depicted by these stripes here. Okay, so they are featured opposite each other, but we'll go into that in more detail in the next slide. But we just need to know that they need to be the same size, the same shape, and the position of the genes must be exactly adjacent to each other. Okay, next, the genes coding for the same characteristic. So what do they mean about the genes coding for the same characteristic? What they mean there is that you can't have the gene for eye color lying next to the gene for hair texture. You can't have it that way. In homologous chromosomes, they need to be found side by side and lying next to each other, and they need to be identical. Okay? And then the location of the centromere. Can you see the centromere is in line with each other there? They are in line. So the one centromere can't be on top and then the other one at the bottom. No. Then it would not be a homologous chromosome. So you might get in the exam a possible example where they give you two chromosomes and ask you, are they homologous? Then you're going to apply your little rule here of shape, size, position, genes coding for the same characteristic, the location of the centromere, and you're going to apply it to the diagram, and then you're going to be able to tell whether it's homologous or not. There are two chromosomes of each kind, one from the mother and one from the father. We know this from our work on DNA, that we have a maternal chromosome and a paternal chromosome. That is why we have some features where when we go out, people will say, oh, you look just like your mom, you have your mom's nose, you know, things like that. Those are the features which we get from our mother and our father, the similar genes that we talk about. So now we're going to discuss replicated and unreplicated chromosomes. Okay, so if you look at the diagram, it's very, very easy to understand. On the left of the diagram, you got the DNA structure. Okay, so this DNA is an unreplicated chromosome. It's found in a single. It is found in a single structure, and you can see the location of the genes there. And what happens? Unreplicated, that means it's on its own. It hasn't made a copy of itself just yet. Okay? And so this is an unreplicated chromosome. If you have to be asked for the definition, it would be the appearance of the chromosome before DNA replication. Okay? And then, if we move to the right of the diagram, this is the example of a replicated chromosome. And this obviously occurs after DNA replication. Very, very simple to understand because we got a singular structure. The structure makes a copy of itself. And if you look at the diagram, look at the position of we'll call it an A gene because they haven't assigned the name of the gene. So look at the position of the A gene. They are in line with each other. The position of the B gene. Okay? So that means, what can we say? It's made like sort of a carbon copy of itself. Okay? And it's identical. So now we know the difference between a unreplicated chromosome, it's on its own, and a replicated chromosome, it makes an identical copy of itself. Right, terminology, very, very important. Terminology is very, very important in life sciences because very often in the exam, when marking the exam, markers pick up that even though a learner knows the idea, they have an understanding of what the concept means. Because they don't use the correct terminology, they're unable to get maximum marks. So please, learners, when you are busy learning your work, when you're busy learning terminology, practice. Write the terminology down and then take another book and then mark it. Mark it in accordance with what you have in the textbook. Use Mind the Gap. Because Mind the Gap is a textbook that is very, very easy to understand. And it's also aimed at making learning simpler for life science learners. And examiners look at Mind the Gap. So if you're looking for simple definitions, easy to understand, clear diagrams, go to Mind the Gap. It's going to really, really help you a lot. Okay? So let's go to our terminology. So we've already discussed this terminology. And now what we're going to do is we're going to read it out loud. That's also another study tip. To read the definition out loud, hear it and understand it. Very often when you read silently, it doesn't always go through. It's like listening to music. Okay? Let's look at terminology. So firstly, we have the unreplicated chromosome. And we already know the definition for the unreplicated chromosome. We know that it refers to the chromosome as it appears before DNA replication. Remember, we learned this definition now. So, we're going to picture it in our mind. We're going to picture that single structure, and we're going to know that that is an unreplicated chromosome. So already when we're picturing an unreplicated chromosome in our mind, we can already know, oh, this one is going to replicate, and then we're going to be able to picture that replicated chromosome aside from the unreplicated chromosome. So let's go to the replicated chromosome definition. This refers to a chromosome as it appears after DNA replication. Because of DNA replication, all chromosome material is doubled. Hence, each replicated chromosome is made up of 2 chromatids, which is joined by a centromere. So it's basically your unreplicated chromosome is going to be, it's going to be a single structure. And your replicated chromosome, as the definition says, it's going to be made up of 2 chromatids. There's your 2 chromatids, and it's going to be joined by a centromere. Do you see how learning with diagrams can make things so much easier? So you should try it. Learn with diagrams. It's going to help you to remember the content. Okay? And then the chromatid. This refers to each of the 2 threads of the replicated chromosome. So there's the replicated chromosome. So that would be the chromatid. Each of these would be the chromatid. It's the 2 threads, isn't it? Right. And then the daughter chromosome. Now the daughter chromosome is something that we've come across. You're probably wondering, what's this daughter chromosome? When you were in grade 10, you did anaphase. And anaphase, remember we said anaphase is a process where the chromatids, the spindle fibers shorten, and the spindle fibers contract, and they shorten, and they pull the chromatids apart to the separate ends, the separate poles of the cell. So the daughter chromosome is exactly what this is. It's each chromatid after it splits from its sister chromatid. Okay, so this is what happens during anaphase. It happens during anaphase. So there's the daughter chromosome, there's the chromatid, the replicated chromosome, and the unreplicated chromosome. So during anaphase, when the chromatid is moving towards a pole, it looks something like that. Do you remember that? And it's like moving this way. So we can picture it as flying away, A for away, isn't it? Okay, so let's move on. Now I've got a beautiful diagram for you. So I want you to look at this diagram. So we can see what's happening here. And while you're looking at this diagram, I'm going to read the definition of meiosis. Meiosis is a type of cell division that results in 4 daughter cells, each with half the number of chromosomes as the parent cell. So can you see what's happening there? That is actually the process of meiosis. Now the process of meiosis involves other processes like cytokinesis, where there is division in the cytoplasm, and then it moves apart, and it also involves karyokinesis. Now karyokinesis is the division of the nucleus into 2 separate nuclei. But that is something we'll come to a little later on. For all you need to know now is that the process of meiosis is a reduction division. It's from one parent cell, it divides to form 4 other cells. Okay, so if we move on to each process, so we're going to start with the phases. Okay, we're going to discuss each phase. Now, what is a problem for grade 12s in the exams is sometimes they know what prophase is. They know that prophase is where the chromosomes, they become distinguishable, that we can actually see them. But then they get confused because they don't know, is this prophase 1? Is it prophase 2? I don't know what I'm doing. What do I do? So I'm going to teach you a trick as to how to remember which one is prophase 1 and which one is prophase 2. And we're going to do the same with the other phases. And what we're going to do now is we're going to draw diagrams. So after this lesson, you're going to draw these diagrams and you're going to put them up in your room, and you're not going to have nightmares. I promise you, it's going to help you to remember. Okay, so let me go through the diagrams and then you can draw your diagrams after this. So if you look at prophase 1, so here you see the spindle fibers are starting to form. Now, very important here, homologous pairs, homologous pairs come to lie next to each other. Can you see there? The homologous pairs are coming to lie next to each other. There we go. There's all our homologous pairs, and they're coming to lie next to each other. Why are they coming to lie next to each other? Because crossing over is going to occur. So when they come lie next to each other, these homologous pairs, they make contact with each other. And when they do make contact with each other as bivalents, crossing over occurs. So crossing over occurs at the chiasmata. Okay, so let's look at this at the bottom of the screen. So here we have the two homologous pairs. There we have the two homologous pairs. They come to lie next to each other. And then do you see what's happening here? The bivalents are actually making contact with each other. There we go. There's the bivalents. They're making contact with each other. Now the point at which they touch is called the chiasma. Don't forget that word. They like to ask that terminology. So whether it's in give the correct biological terminology, or multiple choice questions, or even in answering diagrams on meiosis, they like to throw that sometimes in about the chiasma. So the point of the contact is called the chiasma. And you know what happens? These are called, now when they lie next to each other, we need to be able to distinguish them. So we call them non-sister chromosomes, okay? This one is a non-sister chromosomes, because we can't have sisters bonding. No, that can't happen, okay? So we call them non-sister chromosomes. So these non-sister chromosomes, they come to lie next to each other, and the adjacent chromatid of the non-sister chromatid touches at the chiasma, and part of that chromosome it actually detaches, and it becomes attached to the non-sister chromosome, and it becomes part of it. So if you're looking at this diagram and you want to reference it, the pink actually becomes part of the purple, and the purple becomes part of the pink, as we can see in that diagram. Okay, do you see that? So this is crossing over. Now this is a very, very important process, because when you are crossing over, can you see they're taking some, and then it's becoming part of a new chromosome? What actually is happening there is that they are exchanging genetic material. This is why you don't look identical to your parents, okay? Because the genetic material is being exchanged, and there is variety, and there's genetic variation, so that yes, we still look like human beings, but we differ as the generation goes on, okay? That's how you come to become your beautiful self. Right. So let's move on to prophase 2. So now that we know what happens in prophase 1, we know, okay, when I come to prophase 1, I must look for crossing over, because that's what's going to happen there, okay? Now prophase 2, how are you going to know that this is prophase 2? Because can you see the diagram? We're gonna work with diagrams here, because diagrams is something that we can relate to. So when you come to prophase 2, if you look at this diagram, if I look at this diagram here, okay, can you see this? It's a little patchy. So where did that came from? It came from crossing over. So is crossing over still going to occur here in prophase 2? No. If you are getting a question like this, the examiner is doing you a favor, because they're actually giving you the answer in the question, in the diagram. So look at the diagrams, read them carefully, read the instructions, know what they're asking, okay? So let us read the other things that happen here. The nuclear membrane and the nucleolus disintegrate. Chromosomes have a patched appearance, and that is the difference between prophase 1 and prophase 2. So in prophase 1, crossing over occurs, chiasma. And in prophase 2, no crossing over occurs, it's patchy, and the crossing over has already occurred. Okay? So what is the reason? Now, when we move on, you're going to get exam questions. When you go to into the prelim and in all the tests, they're going to ask you, they like to ask you, which phase does crossing over occur? And then you're going to say, I don't know, I'm thinking. And then you must think of our mnemonic, IPMAT. It can't be interphase, because there's no network of chromatin network there. It can't be metaphase, because there's nothing here in the middle. It can't be anaphase because nobody's moving away anywhere. It can't be telophase because there's no two cells. It has to be prophase. So it's prophase 1. You must say it happens in prophase 1. You can't say prophase 2 because patchy appearance here. Okay? They're going to mark you wrong. So I hope you know the difference between prophase 1 and prophase 2 now. There we have a big sign that says crossing over causes genetic variation. So crossing over is one of those things that cause genetic variation. But there's something else that causes genetic variation. So you have to keep watching to find out what that is. So let's move on to the next slide. Okay, so now we're going to do a summary of crossing over. Okay, let's see if we can do it together. Before the homologous chromosomes come to lie next to each other, and then the adjacent chromatid of the non-sister chromosome makes contact at the, what is this called? Can you remember? Chiasma. At the chiasma, and crossing over occurs, and there you have a patchy appearance which we would see in prophase 2. Yeah. So let us read what we said here on the side. A homologous pair of chromosomes are known as bivalents. Now, bivalence is a term that you should know. Why? Because they will refer to it in the paper. Then you're going to think, what are they talking about? It's only, they are known as, they are a homologous pair of chromosomes which are known only when they are crossing over. Okay? At this stage, they function as one unit and they are connected to each other. So what we're going to do now is I'm going to give you these questions to go through, but we're going to take a 2-minute break, and then we're going to get back to the questions and to the last part of our section. Welcome back, grade 12 life science learners. I'm so excited to be spending the afternoon with you today. We're going to move on to activity 1, and we're going to do a few questions. But what I would like for you to do is, I want you to watch all the recordings again. I want you to make notes as you are watching the recordings and you're making notes, because you're not going to remember everything all the time. We are only human. So even when I'm going through the questions and answers, make notes somewhere. Then when you are studying, you can refer back to those notes and say, oh, I remember the lady said this. So it's important to always make notes, use a highlighter, always refer, have a notebook in front of you, even when you're watching these lessons, so that you can remember this. Because as much as I would love to sit next to you just before the final exam, I'm not going to be there. So you can have my notes with you. So let's continue with activity. Now, there's a lot of grade 12 life science learners, they study their work. They really, really study. But the mistake they make is that they don't go through past year papers. And it's important to go through past year papers. I'm sure you hear your teacher nagging you at school and saying, do this past year paper, do this. They're saying that for a reason. You need to learn in what way they ask those questions. You need to know how they ask those questions. So we're going to do a few of those questions, and then we're going to continue with the rest of our work. And then you'll see now that it can be very, very tricky. So you need to read the question carefully. You can highlight important points in the question if you want to, and then go back and think carefully before you write. So we have a diagram here. We have 3 diagrams here. And in this diagram, we got something going on. I'm not going to tell you because then I'll be giving you the answer. So let's read 1.1. How many pairs of chromosomes occur in a normal human cell? Can I say 46? Can I say 46? No, I can't. Because they are asking how many pairs. So when we are answering, we need to be very, very careful because those examiners can be very tricky. Okay, so we one up on them. Right. There we go. 23, we're right. Okay, so that's how we're going to do the questions and answers. And you must do this in the same way. And it's fine if you do the past year papers and if you get it wrong. It's fine if you make a mistake. Because rather you be corrected now than that marker at the end of the year in the marking center who's going to mark it wrong and then there's no going back. So make the mistakes now. Make the mistakes now, and I promise you, you're never going to make those same mistakes again in the exam. Okay? So let's look at 1.2. So 1.2 says, give labels for structure X. So there's structure X. I'd be very, very worried about you if you didn't know what was structure X. So structure X is the centromere, correct. So structure X is the centromere. And we know that the centromere joins the chromosomes. And we know the centromere is that part that splits during anaphase. We know we have a lot of reference to centromere. So because of those different things, they can put them in a lot of diagrams. So you need to be able to understand what it looks like, where it's found. Okay? Area Y, this process here, they ask you, give labels. Did they ask you what is the process? No, they did not. So please don't go and put the process there. Don't be so excited. Oh, I know it's chiasma and all of that. No, no, no. You need to read the question carefully because then you're going to make a big mistake. So give the labels. So the label for that one would be the, chiasma. And plural is chiasmata, okay? So don't go and write the process there. Read the question carefully. Don't make mistakes. Okay, you can make mistakes now, but not at the end of the year. Name the organ in the human female where meiosis occurs. Name the organ in the human female where meiosis occurs. Now, in the beginning of this lesson, we discussed meiosis takes place in plants and in animals. And we said in animals, it takes place in the male in the testes and in the female in the ovaries. Very good. So this takes place in the ovary. So I have to admit a little mistake I made. When I read this question, I saw ovary, I'm thinking, but how do they know ovary? I myself didn't read the question properly because they said human female. Do you see how tricky it can be? It can be so, so tricky. So now that you're aware and I told you all the secrets, you can read the question carefully and you can be one up on the examiner. Okay? So I think we did well there. Let's move on to the next one. So now they ask you, name, here's it. You're waiting for this, weren't you? Name the process that's occurring in diagram B. Okay, name the process. So the process, we know it's crossing over, isn't it? So we will put crossing over. I mean, look at it. The diagram was already telling you there's a cross, and you just add over, crossing over. You must remember that. Okay, and then you have phase represented by the diagrams above. What phase? What phase is this happening? This whole process, what phase is it happening? Like I said, you eliminate. Don't just be quick to write your answer. Think back. Ip-mat. Is it interphase? No. Is it metaphase? Nothing in the middle. Anaphase? Nothing moving away. Telophase? No two cells. It is? Yes. It is prophase 1. If you write prophase, your answer is going to be wrong. It's going to be wrong, and it's happened in past papers where learners write prophase. It's completely wrong because you're not distinguishing between prophase 1 and prophase 2. And remember the patchy chromosomes that we saw in prophase 2? That is not crossing over. So you need to pay attention to the question, and you need to look at your diagram very, very carefully and answer from there. C. The type of cells that would result from the meiosis of this cell. The type of cells that would result from the meiosis of the cells. And the type of cells that would result from meiosis. Now, what is the point of meiosis? The point of meiosis is to produce gametes. Gametes are sex cells. So what are the sex cells that we know? We know them as the sperm cell and the ova, isn't it? So your answer would be ova, gamete, or sex cell. Okay, so you see how the knowledge that you learned from your notes, how the diagrams that you imagined in your mind, you can apply them now to the questions. You don't even have to write a lot of notes. You just need to go with the diagrams. I promise you, meiosis is a shorter section. It's an easier section. Grade 12s, you got this. Okay. Now, we're going back to that question. Remember when we discussed prophase and we said we don't know whether it's prophase 1 or prophase 2. Now we've got the same dilemma. We're in metaphase. Now which one is it? Is it metaphase 1 or metaphase 2 that they are referring to in the question? So now we're going to refer to the diagrams and we're going to see how we're going to distinguish between metaphase 1 and metaphase 2. As it stands, as I'm sitting here, we already know meta is middle. So I'm already picturing in my mind that I'm going to have chromosomes that are aligned at the equator. But we need to get into the specifics of it. So when it comes to metaphase 1, okay, can you see the pairs of chromosomes that are aligning at the equator? Now there's no set pattern. The homologous chromosomes, they align next to each other. Can you see? They are in pairs here. They are in pairs here. So they randomly align. Now this is very, very significant. The fact that they randomly align means that this is going to promote genetic variation. You know, like the lotto. In the lotto, when they're picking all those numbers, you don't know which number is going to come out as the winning number. Same. They're aligning themselves like those balls are mixed up when they're busy calculating the lotto. They're aligning themselves there, and then when they pair, we don't know which one they're going to pair with. So the homologous pairs of chromosomes align at the equator, and there is random arrangement. Remember this. It is definitely a popular question, random arrangement. And the random arrangement is what produces genetic variation. Okay? The fact that they are aligned here, it produces genetic variation, variety of the species, and also most learners think that, you know, it's during anaphase. You know, when they are moving, when the chromatids are moving apart, they think they are under the assumption that that is what causes genetic variation because they're separating. No, it's the crossing over. And it's the random arrangement of the chromosomes at the equator. Okay? So now we know that we've learned 2 words, random arrangement. Random arrangement is assigned in our minds to metaphase 1. Then we got metaphase 2. Very significant difference. Here we also have random arrangement of chromosomes, but they are in a single. Here they are in a double. Okay? Here they are in a single arrangement at the equator. The equator is located at the middle of the cell, and it's also a source of variation. So if you get two diagrams and then already you see M for middle, because the chromosomes are arranged in the middle, you must remember, oh, metaphase 1 is where they are arranged in pairs, and metaphase 2 is where they are arranged singly at the equator. And if you didn't get the message the first time, I'm going to highlight it for you. Just a minute. There we go. Random arrangement of chromosomes causes genetic variation. This is the reason for genetic variation. That means a variety of genes, a different type of genes for the next generation. Okay? Let's move on. So what we've covered, prophase 1, prophase 2, we know the difference between prophase 1 and prophase 2. Prophase 1 crossing over takes place. Prophase 2, we got the patchy appearance, can't be crossing over. And then we came to metaphase. We said double chromosomes randomly arranged at the equator. And we also said that in metaphase 2, they are singly arranged at the equator. So we'll be able to distinguish them. They must just give us those questions. We already know how we're going to distinguish them. We're ready for them. Let's go to anaphase. I like anaphase. Anaphase, ana, apart, 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 apart. It's like they're flying to the opposite ends of the poles. So let's see what's going on here. Now, what's very, very important with anaphase, okay, is that, what's very, very important with anaphase that you need to remember is the spindle fibers contract. Now, you need to use correct biological terminology. What does it mean when something contracts? It's when it pulls. If something is going to, if I've got something here and I pull it apart, it's going to create tension in the middle. So this is what happens here. So the spindle fibers contract and the chromosomes move to opposite poles. So I want you to look at anaphase 1. Let's look at them together. Let's just create a little bit of variety instead of looking at them in isolation. So we got anaphase 1 and then we got anaphase 2. Okay? Spot the difference. You know those diagrams where you spot the difference? You can already see that in anaphase 1, okay, there's the equator, okay, that's the middle section. We can see here, half of the chromosomes are moving to the opposite pole, the other half, not a third, not a quarter, not 1, 2, 3, half. So half of the chromosomes are moving to the opposite ends of the pole. And we can see they are whole chromosomes. They are whole chromosomes, okay? That are moving to the opposite ends of the pole. But in anaphase 2, so here in anaphase 1, the spindle fibers contract. But in anaphase 2, the centromeres split. Remember the centromere? When we were drawing the diagram early on, okay, I'm not the best drawer, but I can draw a centromere. So there's the centromere. Remember the centromere? So that we discussed earlier, it was even in one of our questions. The centromere, it splits. And you know what happens? It splits in the center, and what happens is the chromatids move to opposite ends of the pole. Can you see that? This set is moving here, and this chromatid is moving there. So you can already see that you have two gray on this side, and you have two purple on this side, and it's halved as well. If you look at the diagram, you can see that it's halved. And what we need to remember is that when they get to these poles, okay, when they get to the poles, we have a special name for them. We call them daughter chromosomes. What do we call them? We call them daughter chromosomes. Oh. Okay, I'm just going to gremlins got in here. Okay, so we call them daughter chromosomes. So these ones we call them so we've come across quite a bit of terminology. We've come across replicated, unreplicated. We've come across sister chromosomes. We've come across now daughter chromosomes. So if you learn these definitions with a diagram, so when you're studying, draw an anaphase diagram and then label it daughter chromosome. Label the spindle fibers in anaphase 1, label them contracting and all of those things. So you don't have to study written notes. You can do the diagram and make your notes on the diagram so that you learn the content and the diagrams at the same time. Because in the exam, they're going to give you a diagram on meiosis. They like to give you a diagram on meiosis. So you need to know the labels. You need to know which phase is which phase. You need to know daughter, sister, replicated, unreplicated. You need to be able to distinguish between those. And that's not going to be a problem for you after today, because now today you've got this. You've been making notes. You're very good. You've got all of these points in your notes. Okay. So we're going to move on now to telophase. So telophase 2. Okay. Telophase 2. So you're going to remember me in the exam. When you see telophase in your mind, you're going to say 2. So basically what happens here, the cell is going to split into, yes, it's going to split into 2. So here we go. So in telophase 2, you have the nuclear membrane reforming. Can you see there's the nuclear membrane? It's reforming. So the nuclear membrane reforms, the chromosomes are replicated, and the cell membrane constricts. Now constricts means it is moving inward. Okay, so can you see there? There's the cell membrane, it's constricting, and eventually it's going to meet here. Okay, and then cytokinesis occurs. This term cytokinesis, you need to know what it is. Cytokinesis is the division of the cytoplasm into 2. Simple. Division of the cytoplasm into 2. Like we did the other term. What was it? Karyokinesis. Yes. But in any case, cytokinesis is the splitting of the cytoplasm into 2. And then if you look at telophase, it has single-stranded or unreplicated chromosomes. There we see them there. Single-stranded or unreplicated chromosome. Each daughter cell has half the number of chromosomes. 4 haploid daughters are genetically different. That is what I was waiting for. 4 haploid daughter cells. We know they're daughter cells. Why? Because they moved to the poles after the centromere split. Okay? And they are genetically different. Now this is different from mitosis, isn't it? Because in mitosis, the one cell made an exact copy. The parent cell made an exact copy of itself. They were identical. But come meiosis, we've got one parent cell. And this one parent cell made 4 genetically different cells. And each of these genetically different cells had half the number of chromosomes as the parent cell. So the parent was diploid and the 4 cells were haploid. Okay? So now we understand the difference between telophase 1 and telophase 2. So we're going to take a short break, and we're going to be joined by my colleague Radine, who is going to continue with the rest of the program. Thank you.
grade 12 learners. Welcome back to the lesson, Life Sciences lesson. My name is Radin Kruger, and I'm going to take you through the rest of the topic that we're going to look at, or that we're busy looking at today. Okay, so we're going to now have a look and quickly have a look at the difference between mitosis and meiosis. You don't need to study mitosis again in grade 12, but they can ask you, sometimes the examiners like to ask, what is the difference? So if we have a look at these two diagrams, if we see meiosis, it says there, meiosis makes distinct gametes. Now, distinct means that they are different. Each one looks as its own identity. So if we have a look down the bottom here, so we started off with one cell, and it went through the first division and it created two cells, and already we can see the cells are slightly different. Okay, and then after the second division we end up with four. If we have a look at them, each one, there's not one that looks the same as the other, so we usually talk about them as genetically different or genetically non-identical. Okay, so that is meiosis. If we look at mitosis, basically we're cloning body cells, and I'm sure you all know the term clone. Sometimes when we've got lots of things happening, we'd like to clone ourselves, that we can then send the clone to go and do something that maybe we don't like to do. So in mitosis, we're basically making new cells. So it's a form of growth, it's a form of repair, it's a form of asexual reproduction. So we end up with two exactly the same. We started out with a cell, a single cell, and it looks, the cells that are formed at the end of one mitotic division or one cell division, they are all identical. So they are genetically identical. We also started off with a diploid cell in meiosis, and we ended up with 4 haploid cells. In mitosis, we start off with a diploid cell, and we end up with diploid cells. Okay, so that's the basic difference. There are other, if we go into more detail, and there are some others, but even what we usually do is then we compare meiosis 1 and meiosis 2. Because if you look at the process of meiosis 2, it's very similar to mitosis. So here we're going to have a look at the differences between meiosis 1 and meiosis 2. But remember, meiosis 2 could just as well be mitosis. So what happens in meiosis 2 is the same as what is happening in mitosis. Alright, so some more differences, some more when we're looking a little bit deeper in. So we have a look, we can see in meiosis 1, the chromosomes arrange themselves at the equator, so in metaphase 1, in homologous pairs. Whereas in meiosis, they line up at the cell individually, which is exactly the same as mitosis. In meiosis 1, whole chromosomes, so in anaphase, when the chromosomes, the homologous pairs of chromosomes separate, it's whole chromosomes that are going to move to opposite poles. Whereas in meiosis 2, no more homologous chromosomes. Okay, so they were, but now it's separated. So now in each cell we've got a single chromosome which has 2 chromatids still, and what happens there is the centromere is going to divide and your chromatids are going to separate. Okay. In meiosis 1 we have the formation of 2 cells at the end of the division, whereas in meiosis 2 we have 4 cells. In meiosis 1 the chromosome number is halved. Okay, so already as soon as we separate those homologous chromosomes and they go to separate cells, I now no longer have pairs of chromosomes. So I've already reduced it from diploid to haploid. Alright, so it's already gone from diploid to haploid. Whereas in meiosis 2, I'm going to keep the chromosomes already haploid. I don't further divide and reduce the chromosomes anymore. So they remain haploid. And then in meiosis 1, it's during prophase 1 that crossing over occurs, whereas in prophase 2 there is no crossing over because we don't have homologous chromosomes. Crossing over can only take place between homologous chromosomes. Okay. Right, so let's have a look at this quick activity. So you can see we can even get meiosis questions and diagrams in multiple choice questions. And this one says, the diagram below represents the structures of chromosomes at different stages of meiotic cell division. Sorry. Okay, so we have a look at the diagram, and we can see there's 4 different diagrams that they've given us. Sorry, can you just take a break?
The road to success is to commit to yourself. In order for you to become who you want to become as a student, you need to commit to reading your books, to studying, to bettering yourself. I know it's not going to be easy. Actually, no one said it's going to be easy. But if you put in the work, the results will show in your life. Tap into your power. Sometimes you will feel like putting away your books, and not reading as much as you're supposed to be. But I want you to commit, because through commitment, there is infinite power. Believe in yourself. Believe a little more in yourself, because to those that believe, nothing is impossible. You have done this before. You have passed your tests before. You have went into a higher grade before. It is possible because you have done it before. Do not doubt yourself. Keep believing in yourself. Keep pushing, because this can be done and you can be great.
So in the diagram, we can see there's 4 different scenarios that they give us. There's Q, which is a pair of homologous chromosomes. There's R, which is a single replicated chromosome. There's S, which is a single unreplicated chromosome. And then there's T, which is also homologous chromosomes. Now they ask which one of the following chromosomes would be found in a cell during late anaphase 2. Remember, Samantha said to you the way in which to recognize the difference between anaphase 1 and anaphase 2 is the number of, or how the chromosomes are. So in anaphase 1, we're going to have whole chromosomes moving across. In anaphase 2, we don't have homologous chromosomes in meiosis 2, so we're going to have single chromatids moving. So the answer then would be which one? It would be the single chromatid or unreplicated chromosome that is moving to the opposite poles. So that would, the answer then would be C. Okay, so we are correct. All right, let's move on. Okay, so now, and what I'd like to just remind you, tell you, is that in meiosis, or for meiosis, examiners love to give diagrams. So all of these questions come off past papers, some of them even from last year. So and we can see examiners like to give the diagram, so you need to recognize the phase that's in the diagram, or you might need to identify certain structures, or you might need to draw a diagram of a phase before or a phase afterwards, or what the gametes are going to look like at the end. So the skill here in meiosis is observation. So you need to look carefully. Look at the diagram and see what is in the diagram. So if we look at these 2 diagrams here, we can see the first one, diagram 1, shows us 4 pairs of chromosomes, homologous pairs, and it looks like they are crossing over. Okay, so we can see the cross. So we see that crossing over is occurring. So we know as soon as there's crossing over, it must be prophase 1. Okay, so if we have a look at the second diagram, we see again there's not single chromosomes, it's homologous pairs, and they're lined up at the equator. So for us, this diagram then must be, we said metaphase is the middle, so that would be metaphase 1 because we still have homologous pairs. All right, so now let's have a look at the questions. So the first question says, name structure B. So let's have a look on the diagram here, and we know this is what, that's considered the pole of the cell. So it would then be the structure that makes up the pole, and that would be the centriole or the centrosome. Okay, so let's have a look and see. Yes, the centriole. Then they ask us to identify structure C, which are the spindle fibers. You can see those little dotted lines. So that would be the spindle fibers. And then the next question, they say identify the phase represented in diagram 1, which we did, and we said it's prophase 1. And then the next question, they say give 3 reasons for your answer to question 1.2. So why did we say that it's prophase 1? So let's have a look. First of all, we said we can see that the chromosomes are arranged in homologous pairs. Secondly, we can see that there are spindle fibers starting to develop around from the centriole. Plus also we see that the nuclear membrane is starting to disappear. Right. And then also the fourth thing that we can see is that crossing over is actually taking place. Okay, so there's lots of evidence here to back up our answer to the previous question. So let's have a look. All right, so first of all, pairing of homologous chromosomes is visible, or bivalents are visible. Secondly, the development of the spindle fibers, as we said. Thirdly, crossing over is taking place. And then the last point is, the centrioles or the centrosome have actually moved to opposite poles. That all happens during prophase 1. All right, and then the last thing, so there's actually 5 things that we can see there, is that the nuclear membrane is disintegrating. Right, let's move on. Okay, so the same diagrams, the next question they ask us is, what is taking place at A? So here they've put a little bracket around and they've labeled that structure A. And so we know where the chromatids are crossing over, that is crossing over actually taking place, the process of crossing over. So now they're asking us to describe the process. So now we have to say what actually happens, how it actually happens. All right, so we have a look. We say parts of the homologous chromosomes overlap because they do. So they now are overlapping. And when they overlap, DNA or genetic material is exchanged. And it's at a point that is called a chiasma if we're talking about one point, but when we're talking about a whole lot, then we talk about chiasmata. Okay, so that's basically where those are the 3 things. All right, then 1.5, they say identify the phase represented in diagram 2. So there we already said earlier that it is metaphase 1 because our chromosomes are arranged here in the middle. And then describe, you see, this is the one where I said examiners can either ask you about meiosis 1 and 2 as differences, or they can replace it and ask you about mitosis. So here they say, describe the difference in events that takes place in the phase mentioned in A. So the phase mentioned in A was metaphase 1, and the same phase during mitosis. All right, so here we can see in metaphase 1, which we said it is, the chromosomes are going to arrange themselves in homologous pairs, whereas in mitosis, the chromosomes arrange themselves singly on the equator. All right, so there we are. In metaphase 1 or meiosis 1, chromosomes are arranged in pairs at the equator, whereas in mitosis, the chromosomes are arranged singly at the equator. Right. And then we're going to have a look at the next diagram. And there we see there's a diagram that says cell P and it's in a phase of meiosis. They don't tell us which one. So there we can see, and if we have a look, we can see the spindle fibers, and then we can see there's 2 chromosomes there. So that would be your chromosome, and then we can see crossing over has taken place. We don't see homologous pairs, so they're going to then ask us questions based on that. So the first question they ask us is, name the phase of meiosis represented in the diagram. So if we have a look at the diagram, we can't see homologous chromosomes, so therefore it would be one of the phases in meiosis 2, and because they are at the middle, then it would be metaphase. Okay, and it would be metaphase 2. Then they say, give the number of chromosomes that were present in the parent cell of cell P. Now remember, this sometimes becomes a difficult question, or learners often think it's a difficult question, but in fact it's not. So if I have 2, this is meiosis 2 already, so if I have 2 chromosomes in the cell during meiosis 2, remember meiosis is called reduction division. So we go from diploid number of chromosomes to haploid. So we half the number. So if I've got 2 in meiosis 2, how many did I have in meiosis 1? I must have, so I double that number and it becomes 4. Okay, so it's actually very easy. So if they ask you about in meiosis 1, doesn't matter which phase, and you count the number of chromosomes and there are 4, then you know in meiosis 2 at the end you're going to end up with 2, because that is the purpose of meiosis. It's to reduce half the number of chromosomes. Okay. Alright, so let's see if we are correct. Alright, so there would be 4. Then they say how many chromosomes would be present in one daughter cell at the end of meiosis. Okay, so we're not going to reduce the number of chromosomes any further than what is here. All that's going to happen is, because remember each chromosome, because it has 2 chromatids, has an extra copy of itself. So the chromosome still has 2 chromatids, but remember we're trying to make 4 cells. So that's why we need to separate them, so that one copy of each of the chromosomes that is left goes to every single cell. So then at the end is going to be exactly the same. It's going to be 2. Alright. Right, then they ask us to name, give the name of structure A. Structure A, basically the bracket goes all the way around the structure, so that would be a chromosome. And then one function of structure B. Now the spindle fibers, they stretch, or they come from the centriole, and what happens is they attach to the centromere of a chromosome. And as soon as that centromere divides, then what happens is the spindle fibers contract. And when they contract, they actually pull the chromatids apart and then pull them to the opposite poles. So their function is to pull the chromatids or the chromosomes to opposite poles. Right, let's move on to the next question. The same diagram. So here they say describe the process. So now once again I need to describe, and examiners very often like to ask this question. Describe the process of crossing over. Okay, so that would result in the appearance of structure A. Right, so when we start out, we talk about chromatids of homologous chromosomes, and they normally are non-sister, so then they will cross over. And when they cross over at that point, the chiasma, those segments are going to break off and reattach to the other chromosome. And that's why you get this kind of patchy look to a chromosome, because it has, so say it's the paternal chromosome, it'll have a little bit of the maternal chromosome on it, and the maternal chromosome would have that same segment would then be paternal genetic material. All right. So during prophase 1, that's when it takes place. Homologous chromosomes lie next to each other, and chromatids from each of them are going to overlap, and where they touch each other, that point is called the chiasma, and it's there that they break off and reattach to the opposite. So we've actually exchanged genetic material, and that is then what we call recombinant DNA. All right. Now, here they've asked us to draw a diagram. So it's still the same. So we originally saw what cell P, that first diagram. So now they say to us, draw a diagram of the phase. So they don't tell us which phase. We've got to work out which phase it is. Occurs after the phase in the diagram of cell P. So now we said diagram, or the first diagram was metaphase 2. So we now have to draw anaphase 2. So if you have a look at the diagram here, so there we must always put in a caption. Remember, whenever we draw a diagram or we do a graph, we always put in a caption. So we must say that's the diagram showing a cell in anaphase 2, and then we're going to actually draw it. And remember, this outer membrane is always the cell membrane. It's not the nuclear membrane. Because remember, in prophase, the nuclear membrane disappears. So that outer circle is the cell membrane. So remember, here's the label. So when you label it, don't label it as the nuclear membrane. Label it as the cell membrane. Okay, so if we have a look at this diagram, we can see, and there's the poles or the centrioles, the spindle fibers. The centromeres have split, and the chromatids are now being pulled apart. So they're actually moving away. All right. Now, the way in which we normally mark diagrams and graphs is we use a rubric. So it's important for you to know what this, or how, or, know what the rubric looks like and how we actually allocate marks. So can you see the first thing is a caption. So the caption is the heading, and there's a mark allocated there. So I must always put a caption, and then I've got 1 mark. Then secondly, the diagram. They've said to us, they're going to look at the position of the chromosomes. So they've got to be in a specific position. 1 large and 1 small unreplicated chromosome or chromatids or daughter chromosomes are being pulled to opposite poles. So they can't be together anymore, because that's not anaphase. All right, remember, apart or away means anaphase. Together is metaphase. All right, and then they say we must show a clear gap between the chromatids. So there must be this gap visible, that we can see that they are moving away. Okay, and then that we get 1 mark for that. And then the correct shading. And that's why I say observation skills are very important when you do this section, because they like to color in and put in a little stripe or the crossing over where it's happened. So you need to color it in exactly the same. You need to shade exactly the same, and then you get a mark. And then any 2 correct labels. So here they've given us quite a few. There are quite a few labels that we can add to the diagram. You only need to give or get 2 correct. Okay, so these kind of questions, a diagram, if you know what you're doing and you know how to do it, then it's actually quite easy marks to get. All right, let's move on to the next activity. Same, now instead of giving us a diagram of cells in meiosis, they've now given us a graph. So already the examiners have kind of raised the level of this question. So let's have a look and see what they say. They say the number of chromosomes in the somatic cells. Now remember, we must know this is terminology, and we must know what these mean. So somatic cells are what we call body cells. So those are all the cells in our body. And remember, all our cells in our body are diploid. So they say the number of chromosomes in somatic cells of organisms differ from species to species. The graph below shows the number of chromosomes in each somatic cell of 3 different species. Okay, so there's lots of information and clues they've put into here. So they've said for each of the species, we can see off the graph. So for giraffe, there's 30 chromosomes, okay, in each cell. For the mouse, there's 40. And for the pineapple plant, there's 50. But remember, they said that it was a diploid cell. So the minute they ask us, okay, so this is 2n, so is that, and so is that. So the minute they ask us for the number of chromosomes in a gamete, we need to halve the number. Because remember, that's what meiosis does. It halves the number of chromosomes. Okay, so let's have a look at the question. How many chromosomes will be present in mouse cells during telophase 2? Okay, so that's the second part. That's where the gametes have already kind of formed. All right, so we have a look here. They tell us a mouse has 40 chromosomes in its somatic cells. So in the gametes, it would be 20. Okay, so let's have a look and see if we're correct. 20, yes. Right, then they say how many chromosomes in a leaf cell of a pineapple plant. Now you learned about plants last year, and you learned that the leaf, its function is photosynthesis, it's not reproduction. So the leaf is not going to be making gametes. Okay, so if they're asking us for a leaf cell of a pineapple, it would be one of the somatic cells. So then I'm going to just have a look on the graph, and I'm going to read it straight off, and that would be 50 chromosomes. Okay. Now, the next question is not a description. They say to us, explain. So I can't describe something. I've got to explain why something is like it is. So they say, explain why the sperm cell of a giraffe has 15 chromosomes. Okay, because we said here that giraffe somatic cells have got 30. Now we need to explain why does the sperm cell only have 15. Okay, so what we need to say is that the germinal epithelium, or the original cell of in meiosis, has got 30 cells, but it undergoes meiosis. And by undergoing meiosis, it's going to reduce the number of chromosomes, and therefore it will end up with 50. So let's see, how did they word it on the memo. So firstly, we need to, because we're explaining. So they're saying a sperm cell is a gamete. As soon as a cell is a gamete, it now no longer is diploid, it is haploid. And that gamete was formed by meiosis and must be haploid. And the reason why it must be haploid is because the sperm cell on its own is not going to become an offspring or an organism. It needs an ovum, okay, another gamete that it must fuse with. So if I put 15 and 15 together, when they fuse in fertilization, it's going to go back to the original number, which is 30. Okay, if I don't, I'm then going to create, or the chromosome numbers are going to change, and then the structure is not going to develop. All right, so that's when we have an explanation. I can't just describe the process of meiosis, of how it went from a diploid cell to a haploid cell. I've got to say why. Why is it haploid? Why does it have 15? Because it is a gamete, and gametes are formed through meiosis, and when they are formed through meiosis, then they become haploid. All right, let's have a look at some more of the questions here. The next question, they say, name the phase of meiosis where the halving of the chromosome numbers begin. So remember, we said in meiosis 1, in metaphase, my homologous chromosomes are going to be lined up next to each other in metaphase. Where do we start separating those chromosomes? We started in anaphase 1. So already in anaphase 1, my chromosomes are separating, and they're now going to form around each of the poles, and then once constriction or cytokinesis has taken place, we're going to end up with haploid cells. So the process begins already in anaphase 1. Right. Then we need to describe the events in the phase. So now they're asking for a description. So now I must say what actually happens during anaphase 1. So that was the answer to question 3. So I say my spindle fibers shorten. So remember I said to you earlier, that's what happens. The spindle fibers contract and your homologous chromosomes, or actually it's those. Now what happens in anaphase 1, your homologous chromosomes now going to be pulled apart because the spindle fibers are shortening. It's like elastic and it's pulling the chromosomes towards the poles. So the spindle fibers shorten or they contract, the chromosome pairs separate and they move to opposite poles. That is a description of what happened in anaphase 1. Alright, let's move on to the next activity. Okay, so here again they've given us a diagram showing different stages during meiosis. And if I just look at diagram 1, I can see there are still 2 chromatids that are joined by the centromere in each one of the chromosomes. So that possibly, they are replicated chromosomes. So for me that already indicates that it's probably meiosis 1. Secondly, I can see that there's a gap here between the chromosomes. So that means that they are moving away to opposite poles. So therefore it would be anaphase 1. If I look at diagram 2, here I don't see 2 chromatids forming a chromosome. Here I just see a single chromosome, what we call an unreplicated chromosome. I also see that something's happening here. So I look like I'm getting 2 cells. So I can see here constriction is happening and eventually it's going to constrict completely and the 2 cells are going to separate. So this is cytokinesis taking place, and because it's single unreplicated chromosomes, this would then be telophase 2. Okay, so that's why it's so important that you do know, you are able to identify the phases, and there are lots of clues and that you need to learn. Alright, so the first question they ask us is identify part A. So they like to ask this question because we often confuse the centriole with the centromere. Okay, and remember the centromere is what joins 2 chromatids together and the centriole is what forms the pole. Alright, so part A would then be our centriole, or we can say the centrosome. Okay, so they will accept that. Identify the phase. Okay, so I think that half the question has disappeared. Let's see if we can get the rest of the question. Okay, so in diagram 1, I would assume that question is because it is anaphase 1, which we said we already had a look. And then they say describe the events that took place in the phase before the one represented in diagram 2. Okay, so there we're looking at anaphase 2. And so what happened there is the centromere divided and the spindle fibers are going to contract. So the examiners like anaphase, whether it be anaphase 1 or 2. Spindle fibers contract, the centromeres have split, and then the chromosomes are pulled to opposite poles. Alright, let's have a look at some more questions. Okay, name the process that causes the chromosomes to have a combination of genes. So here we can see, here's a combination. We can see they've got, remember I said it's that kind of patchy look even here. You can have a look and see. We've got differences. So what is the process that created that recombinant DNA or allowed the chromosomes to exchange genetic material? And that would be, remember, it's that process where the cross is, that is called crossing over. Okay, so we've got crossing over. Then they say, give one reason why the process named in question 4 above is important. And the reason is as soon as we do a bit of crossing over, is then we've got recombinant DNA, and what are we doing? We are creating variation, so that every single organism that is born is not identical to the one before. So the gene pool gets wider. And in fact, the importance of that, the significance of genetic variation, is it allows species to survive better, gives them better survival ability, a population, if not every single individual. Because as soon as, say, we get some kind of a disease and everybody has exactly the same genetics, then if one dies, they're all going to die. Okay, so that is why crossing over is so important. All right, then they say, okay, so that was it. It's because it leads to genetic variation. Now, here, if we look at the diagram, we see there's only 4 chromosomes to start off with, in the first cell. But now they're telling us, if this was a human cell, how many chromosomes would be present in the cell during the phase represented in diagram 1? So there would still be all the chromosomes would still be there, which means there would be 23 pairs, or there would be 46 chromosomes. So we just can say 46 chromosomes, okay, because they're asking us how many. They didn't say how many pairs. And then the last question, they say structure B and C are both chromosomes. Explain why they are structurally different. So now I have to look. I can see B consists of 2 chromatids with a centromere keeping them together, and C is a single unreplicated chromosome. So I now need to explain why they are structurally different. Okay, and so we have a look. Structure B, we said, consists of 2 DNA molecules, whereas structure C only consists of 1. And that's, so structure B has 2 parts because of DNA replication, whereas structure C consists of only 1 DNA molecule because it is unreplicated. So they separated, and it hasn't gone through replication again. So they remain single molecules. Okay, and that is as a result of the splitting of the centromere. Okay, right. Then, if we have a look at this question here, again, they've given us a diagram, and they say identify this. First of all, they say the figure below represents a cell in a phase during meiosis 1. So they've been kind and they've told you at least it's meiosis 1. All right, then they say identify the stage of meiosis shown. So if I have a look, to me those chromosomes are lined in the middle on the equator, and they are occurring in pairs. So I would say that would be metaphase 1. Right, then they say give a visible reason, and that's what we just stated now. We said that the homologous pairs are lined up, so the chromosomes are in homologous pairs along the equator. And then they ask us to provide labels for structures 1 and 2. So 1 is indicating both of these chromosomes, so that would be a homologous pair of chromosomes. And then 2 is just indicating a single chromosome, but they're pointing to the 2 chromatids. Okay, so 1 is homologous chromosomes, and 2 then is sister chromatids. Right. And then they say, would you consider the cell shown in the figure to be haploid or diploid? As soon as I've got pairs of chromosomes, it's not haploid, because I've got 2 of each. All right, so I don't only have 1 chromosome, I've got 2, and that means that they would be diploid. All right. Okay, so it would be diploid. Explain your answer. Okay, we said that because there are still homologous chromosomes there, means that the chromosomes are still occurring in pairs, and therefore it is a diploid cell. Right, so homologous pairs have not yet been separated, or they are still present. And then how many chromosomes would be present in each of the daughter cells? So remember I said to you, if you look at the cell, and if it's in meiosis 1, so there we can see there's 4 chromosomes, and in meiosis we're going to half the number of chromosomes. So we're going to end up with 2 chromosomes in each of the cells formed. Okay, so it's going to be 2. All right, now let's just quickly look at the importance of meiosis. I alluded to it just now when I said to you, imagine if everybody in a population had all identical genetic material, so that as soon as environmental conditions become unfavorable, everybody will die, and then the population doesn't survive. So if we have a look here, we can see some of the importances. So the first thing in reproduction, so we have in these 2 ducks, we have the male and the female, and what we see is that they have 80 chromosomes. So if I put 80 and 80 together, I'm going to get 160. And that's not the duck anymore, because that duck only has 80 chromosomes. So what has to happen is the number of chromosomes need to go from 2n to n, and that is 40 chromosomes. So that when I put 40 and 40 together during fertilization, so when I add them together, I'm then going to end up with 80. So one of the importances of meiosis is it maintains the chromosome number, or it creates haploid gametes. Okay. Then another one is that it creates genetic variation. So the first thing, it produces haploid gametes, or it maintains the chromosome number. Then the halving effect of meiosis is overcome, okay, by the doubling effect of fertilization. So as soon as I've got those haploid chromosomes during fertilization, they go into the cells. I'm going to join them, and then it goes back to diploid. So the number of chromosomes stays the same. Right, so exam tip, and that is why Samantha also said earlier, is we must look at the question very carefully. Because if we look at organisms, different organisms, their fruit fly only has 8 chromosomes, whereas humans have 46, as we've said. A rat has 42. A fen duck, which is the one we were talking before, had 80. Tobacco has 48. Barley has 14. Watermelon has 22. And a bean has 22. So when we look at the diagrams that the examiners have given us, we have to look to see, did they say it's a human cell, or did they say it's just a random cell? If they want us to interpret it as a human cell, they would say, in humans, how many? Okay, so that's what you need to look. You need to read those questions very carefully. All right, so read the question carefully to see whether examiners are referring to humans or other organisms, and whether the cells are either somatic cells or gametes, because that would also make a difference. All right, then the second significance of meiosis is that, as I said, it introduces genetic variation. And there's 2 ways in which it does it. The first way is through crossing over. Okay, and there we see the diagram again, and here we can see where the crossing over is actually taking place. And what happens is we end up with genetically different. So there the genetic material has been exchanged. All right, so that's one way. And the second way is through random arrangement. When the chromosomes line up at the equator, there isn't a recipe that says all the paternal chromosomes must be this side and all the maternal chromosomes that side. It happens randomly, exactly like the lotto. All right, so there if we have a look, we can see there's different possibilities, and they've just shown 2 possibilities here, where we see how the chromosomes have lined up, and we have a look at the gametes at the end. And possibility 2, we have a look. So now, just out of these 2 possibilities, we end up with 4 different combinations of gametes. So can you see how it creates genetic variation? All right. Now, here, this is another kind of diagram that they give us, where they show us what we call a karyotype. So this diagram is called a karyotype. I'm going to write it here. Because what we see is every single chromosome, and because there's 23 pairs here, we assume it's in humans. They also tell us it's in human somatic cells, and we can see that all the pairs of chromosomes are diagrammatically displayed in order. So you can see them. Right, then the first question they ask us is, what is a somatic cell, or what are somatic cells? And we said somatic cells are our body cells. So they are body cells, or they're all the cells in the body except the sex cells or the gametes. Then the second question, they say, name the specific type of chromosomes numbered 1 to 22. So that's the last pair, okay? We call those gonosomes because they are the sex cells, a sex chromosome. Now all the others, from pair 1 to right up to 22, they are known as the autosomes. Okay, they have nothing to do with determining sex or the gender of the individual. So those are known as the autosomes. And then they say each of the pairs shown is a homologous pair of chromosomes. Okay, so there we can see there's 2, and you can see they are more or less the same size, same shape. The centromere is in the same position. So each pair looks pretty much the same. Right, now they say, state the origin. So where did one of the homologous pair come from, and where did the other one come from? All right, so that would be one came from the sperm cell and one came from the ovum, or one came from the father and one came from the mother. All right, so one is paternal in origin and the other one is maternal. All right, let's have a look at the next question. Okay, so here they say the diagram represents the chromosomes from human somatic cells of 2 individuals who are twins. But if I have a look over here at pair 23, that one tells me it's male because there's an X and a Y chromosome, and this one tells me it is female. So they wouldn't be identical twins. All right, but let's have a look at the questions. Each of the pairs shown is a homologous pair of chromosomes. List 3 characteristics that chromosomes in a homologous pair have in common. So remember we said they're the same size, the same shape, the centromere would be in the same position, they carry exactly the same genes. Okay, so it might be different alleles or different forms of the gene, but the gene, if it's for eye color, then it would be in the same place on both of the chromosomes. So let's have a look at some of the characteristics. So the shape is the same, similar, and the same size or the same length. The position of the genes, as I said, would be in the same position, same locus. They carry exactly the same genes that code for a specific characteristic, and the location of the centromere would be the same. All right, so any of those. Then the fourth question, they say, explain one observable reason why the 2 individuals are not identical twins. And remember, when we first looked at this diagram here, we said if we have a look at the gonosomes or the sex chromosomes, we can see the X chromosome is much larger than the Y chromosome. So individual 1 would be a male and individual 2 would be a female. And that is why they are not identical twins. All right, so the gonosomes are not identical, or the chromosomes at position 23 are not identical, and therefore individual 1 has an XY gonosomes or is male, and individual 2 has 2 X gonosomes or is female. Right, and then the last activity that we're going to look at for today, again a slightly different kind of question. Here they say the graph alongside shows the varying amounts of DNA in the nucleus of a cell during the cell cycle. So if we have a look here, here they say, so on the y-axis is the amount of DNA, and on the x-axis is over time. And they've numbered the different stages. So here we can see there's a certain amount, and then it doubles. So that at point 2, the DNA is now going to be double the amount of DNA. Then at before point 3, we have the reduction of the amount of DNA. So we go back down to normal, and then we have a further reduction where we go down to half the amount of DNA. Okay, so I'm sure you can already think of some of the questions they're going to ask you. So let's look. The first one. Which type of cell division is illustrated in the graph? So the fact that we start out with a certain amount of DNA and it doubles, that's what happens during interphase, okay? But then at the end, at point 4, we're going to end up with only half the amount. So therefore, this type of cell division must be meiosis. Okay. Then which phase occurs between points 1 and point 2? And that would be interphase. And the way in which we know that is because we said the DNA went from a certain amount and then it doubled. Okay, and that's during DNA replication. So let's look at the next question. They say describe the decrease in DNA content per nucleus between points 2 and 3. So that is when, so if this is meiosis, then we're going to say in meiosis 1, what happens is my homologous chromosomes are going to separate, and then we're going to then have a further division where the actual chromatids separate. Okay, so that's how it reduces. Okay, so between points 2 and 3, we have during meiosis 1, telophase 1, homologous chromosomes are separated, so there's no longer homologous chromosomes. So that's how we've reduced the amount of DNA here from point 2 to point 3. And then between point 3 and point 4, the same thing. During meiosis 2, telophase 2, daughter chromosomes are separated. So now we no longer have a replicated chromosome, they're separated. And so now we have a single or unreplicated chromosome going to each cell. And then, so we've divided the chromosomes into different or 2 nuclei when the cell divides. And so that is how we've gone from a certain or full amount of DNA down to half. Alright, and that is the end of today's lesson. I hope there was a lot of useful information that you could have gathered, and I hope you were taking notes. And remember, you know, you can use these videos right throughout the year when you are revising for your June exam, your prelims, as well as the finals. So good luck, grade 12s, and we'll see you again soon. Thank you.